So our next two presenters are well known for their ability to communicate scientific information in a way that is both accessible and fun. And I'm very privileged to be able to work with them. So up next, I'm very happy to introduce Dr. Rachel Harding and Dr. Maiuri delivering the keynote presentation, HDBuzz, a clinical trial update. Before they dive into the presentation, I'd like to tell you a little bit about them. Dr. Harding is an assistant professor at the Faculty of Pharmacy, principal investigator at the Structural Genomics Consortium, and cross-appointed in the Department of Pharmacology and Toxicology at the University of Toronto. Dr. Harding completed both her undergraduate and graduate studies at the University of Oxford, and moved to the University of Toronto for postdoctoral training, where she was awarded the prestigious Huntington Disease Society of America Herman Topper Family Career Development Fellowship for her research on Huntington disease protein. This work has continued into her independent research career. Her research team now focuses on uncovering the mechanisms driving repeat expansion disorders such as Huntington disease and advancing early phase drug discovery for novel targets. Her contributions to the field of HD were further recognized with her award of the 2024 Nancy Wetzler Young Investigator Prize, an honor bestowed upon a researcher who reflects the highest caliber of excellence, diligence, and creativity. Dr. Maiuri is the Associate Director of Research and Patient Engagement at the Huntington Disease Society of America and a Research Associate in the Truman Lab at McMaster University. Nearly 15 years of experience in Huntington disease research, she is dedicated to bridging the gap between laboratory science and the lived experiences of families affected by Huntington disease. Dr. Maiuri is actively involved in the HD community through her volunteer work with the Huntington Society of Canada, where she also serves on the Research Council, and as a science writer for HDBuzz, a role she has held since 2012. Her work spans scientific research, knowledge translation, and public engagement, all grounded in strong commitment to patient-centered progress. Together, Drs. Harding and Maiuri will share an accessible update on the latest Huntington disease research that highlight progress in clinical trials, new treatment approaches, and important scientific discoveries, all explained in clear, understandable language for you to all have a little fun along the way. Over to you, HDBuzz.
Well, thank you so much for having us here today. We're so delighted to be able to give you the HDBuzz Research Keynote here in Canada, which is, well, both of my adopted home and Tat's home as well, so it's really a super pleasure. So HDBuzz, for those of you who don't know, we're a group of scientists who come together to try and translate some of the latest science, clinical research, and other bits and pieces that are coming out so that it's in as accessible language as possible for folks like you, and also for other scientists and other people along the way as well, so we can all be in touch with the latest news and updates that are going on. Oh, I don't know if it's my this slide's changing. Oh, hang on, I need to go back one. I haven't quite figured out these gloves just yet. Okay, here we go. And so Tam and I have been a part of this for quite a long time now, and yeah, we're delighted to be here with you all. Okay, so I think that's okay. We just have to remember that it's on the slide. That's on the right-hand side. Oh, yeah, just that's okay. We're just this is just obviously a very well organised and not at all clumsy presentation that we're doing today. Okay, so just a little bit about me, just so you know who we are as we're talking here today. So yeah, as Alexander very graciously introduced us, you might also guess from my accent that I'm not originally from Canada. I grew up in the UK, in the town of Dover, where's the White Cliffs and Big Castle, really close to France. And then I did my undergraduate and my PhD at the University of Oxford before moving to Toronto for my postdoc. And I love Canada and I love Toronto so much that I've been here ever since. I'm so lucky to have the opportunity to continue to pursue what I think is important research in Huntington's disease, and I now actually have my own lab at the University of Toronto, We have like 15 folks in the lab who are all dedicated to HD research, which is super awesome. And there's actually, some of them are here today. Actually, they're all down here at this table. Do you guys want to stand up? Megan and Eileen around, you can ask them anything you like about Huntington's disease research, and they'd be happy to talk with you. And so we're really interested in understanding exactly what's going on at a molecular level in Huntington's disease and trying to find new ways that we can develop drugs for HD.
All right, thanks Rachel. My name is Maiuri. I know lots of you. For those of you who don't know me, I'm also from an HD family, like most of you. There's a picture here of my mom and I. We lost my mom 2 years ago to HD, miss her dearly. But it was, I originally did my training in cancer research, and it was when we found out that mom had the gene that I switched fields and went to work with Dr. Truitt at McMaster University. And spent a lot of time there trying to figure out, what the Huntington protein normally does and how that might go wrong in HD. But recently, back in January, a position opened up at the Huntington Disease Society of America, which was perfect because it combined working on HD research, managing HD research projects, but also doing the community interaction part, which I love so much. And so super happy to be at the HDSA now. But long history also with HSC. So my family has been very much connected to HSC for a very long time. This is a picture of my great-grandma Pearl at her 100th birthday with my mom and my Aunt Dorothy. So Grandma Pearl lost her husband and all 4 of her children to HD. Aunt Dorothy, got together with Ariel Walker at the very beginnings of, Ariel and Ralph Walker at the very beginnings of the HSC. And you can see this is a clip of the horizon from 1994. And it's got Grandma Pearl and Ariel and Ralph, and it was when Grandma Pearl passed away at the age of 101. And it says, she will be sorely missed, but Pearl Smith left a legacy, her family, who now carry on the fight against HD. And it makes me very, very happy to be part of that. So Grandma Pearl's legacy involved Aunt Dorothy and Stan Edwards, who established the Niagara chapter. My dad still volunteers for over 40 years now, and this is him receiving an award from the city of Niagara Falls for his work. And my husband and I played in a band called Eli and the Strawman. We raised over $50,000 over the course of a few years of benefits, and time goes on, our kids got a little bigger and we couldn't tour around Canada anymore. And so we're not playing in the band anymore, but we've changed roles a little bit. And so Rachel and I, along with 2 of our other Canadian colleagues, Mahmoud Biladi and Thiago Mestre, we formed the HSC Research Council. So happy to serve in that role where we basically help advise on research-related topics. Okay, so before we get to the HD research update part, we just wanted to give a shout out to Rachel's co-editor-in-chief of HDBuzz, Sarah Hernandez. So Sarah also has a science background. She did her PhD at UC Irvine in California. And she also now works for a nonprofit, which is formerly called the Hereditary Disease Foundation, now called the Huntington's Disease Foundation, which makes a lot more sense, I think. So Sarah, was home with her kiddos for Halloween, and so she couldn't be here today. We're not sure if she's also dressing up as the Wicked Witch of the West, but we'll give a shout out to Sarah. Okay, so Rachel,
Sorry, Sam, I keep getting you every time. Yeah, so in case you haven't already established what the theme of this year's talk might be, we're very happy to welcome you to the world of Oz, but also of HD research. And so just be prepared for lots of very clumsy metaphors and like loosely thrown together analogies, linking all of this in together with some of the latest science and clinical updates as we get into everything today. Okay, so we thought this was a good theme because there are, here we go. All right, sorry, I'm having a little bit of a delay on the old slides. There we go, all good. So for most of us sitting here in this room, we're very aware of the turbulence that something like Huntington's disease can bring to people's lives, how it can uproot people and how it can bring us into a place of uncertainty, of like fear and really not being clear how things are going to move ahead and how things are going to be in the future. But whilst this storm exists, we together are the HD community. We are all focused and determined to get us to what we see as the Emerald City, which is these disease-modifying therapies for Huntington's disease. So what this means is these are kind of the drugs that will either slow, halt, or perhaps even reverse signs and symptoms of Huntington's disease. So all of us together as the community, researchers, families, folks, advocacy organizations, people at HSC, we're all on this yellow brick road together, and this is the aim that we're all heading towards, to have these accessible therapies available for everyone. And so today in our talk, we're going to be covering a few different things. I think most people in this room are aware there's some very hot research updates straight out of the clinic, so we're going to be focusing on some of those. But before we get into that, we're going to give you a bit of background on HD genetics, because I assume there's probably some people in this room who have never been to an HSC meeting before, so we want to make sure we're all on the same page before we dive into the science. And then we want to tell you about some of the really exciting stuff that's not quite in the clinic yet, but people are working so hard towards to give us more and more options to be thinking about and how we might be able to develop drugs for Huntington's. And so to start off with, we're going to get into a bit of a background, some biology 101 on Huntington's disease genetics. So everyone inherits a copy of the Huntington gene, one from their mum and one from their dad. As we have two copies, everyone has two copies. It's just that people with Huntington's disease, one of their copies has just an extra bit of DNA in it, and so it means you have a disease form and this healthy form as well. And what this really looks like, this extra bit of DNA, is there's this region in the DNA that's called the CAG repeat, and people in the room are probably familiar with the CAG number concept, which is what you get when you have a blood test for diagnosis of Huntington's. And so folks who have somewhere in the order of 10 to 26 of their CAG DNA letters over and over again in their Huntington gene, they're going to be healthy, they will not get Huntington's disease in their lifetime. We have these kind of what we call the grey zone, where we have folks who have just a bit more than that, who either their children might develop Huntington's disease, or they may or may not develop it within their lifetime. But typically the kind of definition within the field is that if you have more than 40 of these CAGs, you will definitely develop Huntington's disease at some point in your life. So this is what we refer to as the expanded Huntington gene. And this expanded Huntington gene makes a different form of the protein that's encoded. So instead of making what we call the wild type or regular Huntington protein, we make this expanded version of the Huntington protein. And this is kind of the central dogma or our fundamental understanding of Huntington's disease. And what we know is that in the absence of this expansion, we seem to have very healthy neurons, things seem to be going well, but when we make this expanded form, things, our nerve cells just look a bit sick and things aren't so happy.
So as Rachel explained, this is disease caused by a single gene. But as you will all know from your experience, HD doesn't happen the same way in every person. Even if someone has the same CAG length as someone else, it may come in at different times, it may come out in different ways. And so we're starting to understand this a little bit better now. Sometimes people have the exact same CAG length, but they might get symptoms earlier in life. Another person might get symptoms later in life. And about 10 years ago now, there was a very, very large study that basically asked what other genes in people's bodies might influence whether people get these symptoms early on or later in life. And what they found was that many of these little, very small changes in people's DNA that track with either early or later onset of symptoms, these little changes tend to be in DNA repair genes. And so this has kind of exploded in the field after that very large study. This is a graph from the study. I think the most important part of this graph, if you look and you see in the top left corner and it says N equals 9,009. What that means is that 9,009 people participated in this to get this information. And it's just a testament to the dedication of the HD community to get this information. We wouldn't know that you need a lot of people to be able to find those small changes that track with early onset or late onset. And we wouldn't be able to find it if we didn't have many, many people. In fact, this is the first study, but there have been more, and we know that there's over 20,000 people's DNA has gone in to understand what these little changes do and how they track with earlier onset or later onset. And so the other parts of this graph worth looking at, the words that are in black, those refer to genes that are not DNA repair genes. So while lots of these genes are involved in DNA repair, there's actually still options for us to look at, different leads for us to look at in the other ones. Those in red are DNA repair genes, and those highlighted in yellow all have to do with a special kind of DNA repair called mismatch repair. And so the take-home message here is that we now have many leads to look at things that we know, in nature, symptom onset can be earlier or later. And now we know what things are associated with that. So if we can influence those things, we can influence symptom onset and hopefully push it off. And so a lot of people are looking at these genes that are highlighted in yellow that are part of the mismatch repair, DNA repair pathway, because it's involved in a term, very fancy long name, called somatic instability. You might also hear it called somatic expansion. So let that one sink in. You're going to hear it a lot because there's actually quite a bit of exciting research coming down the line, which we'll talk about. But just to give you an idea of what somatic instability or somatic expansion is all about, in most cells, the CAG length that you're born with stays the same. If you're born with 42 repeats, most of the cells in your body will have 42 repeats in the Huntington gene. But in some cells, mostly in the brain, the CAG repeat can actually get longer because it's unstable. And that's what we call somatic instability or somatic expansion. So where does the DNA repair come in? The mismatch repair. These mismatch repair proteins are actually trying to fix that DNA, but because it's a little bit too long and it forms these funky structures, the mismatch repair genes actually mess it up and start and put more CAGs in. And so the consequence is thought to be that extra long CAGs are causing a storm in those cells of the brain. Clumsy metaphor number one. Get ready for it.
But so what does this mean in terms of if someone with Huntington's disease diagnosis, they get a CAG number in their test result, will this change with somatic instability?
Right, so because this is only happening in a few cells here and there, mostly in the brain, it's not going to change throughout the rest of the body. So if you get a blood test at one point in your life and you go get another blood test 20 years later, it's likely to be the same number. So yeah, it's just happening in those particular brain cells, and that's thought to be what's causing problems in the brain cells. You know, as with most things about HD biology, it's not very straightforward. It's very complex, and the more researchers look into it, the more it seems to get more complicated. But this area of research has exploded over the past decade, and so study after study after study is starting to put all of these pieces together. So it's also brought in researchers from other areas and other domains to come and work on HD. Because they may have worked on somatic expansion in cancer or another neurodegenerative disease. But we want as many people researching HD as possible. So this has made that happen. Many studies are trying to understand, you know, how this somatic instability may drive disease progression. But actually, the truth is, even if we don't figure out exactly how it's working, we know the targets. We know those genes that were associated with people getting symptoms early versus late. And so if we can make drugs against them, hopefully the idea is that we can mimic what we know happens in nature and influence those proteins, those genes, and push it off so that people can get symptoms much later. So the idea is basically creating drugs against these mismatch repair genes and delay symptom onset. And so several companies are doing just that. None of these are in the clinic just yet, but so they're still just testing in cells and mice. But basically, Locus 23, Lattice, and Pfizer, those are three companies that have their site on a protein called MSH3. Harness Therapeutics is the only company I know of that's targeting a protein called FAN1. And then Revere and Argenta are working on another one called PMS1. Why am I giving you all these acronyms? The point is, they're taking multiple approaches. So, you know, we've got multiple approaches to address HD. Targeting somatic instability is just one of them. And even within that, different companies have their sights set on different things. And so the more options we have, the more likely we are to have one that works. So we hope that one day somatic expansion will turn into the good witch and all our dreams will come true. And put Toto down.
Oh yeah, Toto the donkey. Yes. It was hard to find stuffed dog at short notice. Okay, so this is all really exciting, and particularly, you know, having really big pharmaceutical companies like Pfizer interested in developing drugs for Huntington's disease. These are companies that have the runway, the resources to move quickly and get stuff done. So we're so happy that we have these kind of big companies in here, as well as these more innovative, smaller and scrappier biotechs doing some cool stuff as well. But all of this means that we're heading with so many different directions into the clinic, and there's all of these different strategies. And you know, the key thing to remember is we don't just need the scientists and the clinicians and other people to do to get these drugs to the clinic. You know, many people in this room are the people who have made these trials, past, present, and things in the future happen. Like this happens because folks like you participate in research, and this is the only way that we're going to be able to push things forward. So you always have to remember that you are also a part of this process. So Sarah Hernandez actually made this really nice slide, which kind of shows all of the different things that are happening in terms of drug development for Huntington's disease. So we have drugs already available to treat some kinds of symptoms of Huntington's, but what we're mainly going to focus on today is what we call the disease-modifying. And you heard about one type of these disease-modifying therapies, which is all focused on this concept of somatic expansion, the change in some cells in our bodies that leads to the CAG number to get longer and longer. And this is, a really compelling narrative that scientists are trying to chase to see if they can slow this down and then slow down Huntington's disease as well. So we learned a lot about the companies in that space, but I really like this diagram because it shows you all of the companies who are working in all of these different areas of research and so many different ideas and approaches beyond just somatic expansion. It's like it's a busy space. There's a lot going on. And even Dorothy is, pretty shocked with how much work everyone is doing. So probably the approach that most of you are most familiar with here is this idea of Huntington lowering. And you can see on the diagram, this is where most of these companies seem to be focused. And this whole idea is about reducing the amount of the Huntington protein, the disease form of the Huntington protein that we talked about earlier. Like just getting the levels of that just down a bit. And this is, this approach was designed because when we first started designing these therapies 10, 15 years ago, we didn't know about somatic expansion as potentially being something that was driving disease. Now we've got people working on that. But what we knew about 10, 15 years ago is that this is kind of the root cause of Huntington's disease. So maybe if we can target things at the root and reduce the amount of that disease protein, we can move forward. I just realised I'm reading Tam's slides, so I'm stealing your spotlight. I do apologise. The gloves give me the power. It's like, let me take over.
I'm trying to put my heels together. Okay, so I was tasked with explaining about Huntington lowering. So here we go. As Rachel explained earlier, there are two copies. Everyone inherits two copies of the Huntington gene, one from mom, one from dad. And most people with HD have a healthy copy and the disease copy. Ideally, we would like to get rid of the disease copy and keep the good one. It turns out that is not that easy to do, although there are approaches to this. But several companies are taking different approaches that actually lower both copies. And so these different approaches are kind of like the different paths down the Yellow Brick Road to take us to the Emerald City. And luckily, we have several paths. So let's go through them. We're going to start with the companies that are targeting both copies of the Huntington protein. And so we've got Skyhawk, Unicure, which I'm sure you've heard about in the news, and Rachel will give you an update on. Spark and Roche, PTC, which is being taken over by Novartis, and Alnylam. All of these companies are taking different approaches, which again, just like I was saying with the somatic instability, it's even within this one approach, there's several sub-approaches. Lots of shots on goal, okay? This is the point. And so, let's see. So those 6 companies were aiming to lower both copies of the Huntington protein. Vico is a bit unique in that it's preferentially targeting the diseased copy of the protein. So it still lowers the good copy a little bit, but it lowers the diseased copy more. Wave has an entirely different approach, and so what they're doing is targeting a small variation in the Huntington gene that is commonly found just with the diseased copy of the gene, and not the healthy copy. And so by targeting that little variation, they can, it's called being allele specific. So they're specifically lowering that version of the gene.
Do we know which of these approaches is going to be the best?
We don't know, Rachel. I mean, ideally it would be nice to just lower the disease copy, but the truth is we have some trials that have been going on for several years now that lower both copies, and it has been safe so far. So we are going to wait and see. But, maybe one day it will be as easy as smelling magic flowers. But for now, there are 3 main ways that these companies have developed these drugs, and 3 main ways that they're delivered into the body. I like to break them down this way because this is kind of like what's important to what it's going to be like day-to-day life to be treated with these drugs. And so, you know, pill form would be fantastic. It's very easy to do. You know, take a pill every day. It affects the whole body, not just the brain, and so that could be good if the disease protein is, you know, causing harm outside of the brain as well. We also don't know, you know, if that might mean more chances for side effects because we don't know the effects of lowering Huntington in the whole body. So there's a trade-off there. Spinal injections are another way to deliver the treatment more directly to the brain, but of course that would require ongoing procedures. And then gene therapy is a one-time brain surgery. So that's obviously the most invasive, but it might also last the longest. So I hope that, you know, those of you who have been coming to National Conference for a long time will agree that, you know, we've come a long way in the last decade. And so I'll hand it to Rachel to talk about this progress and the latest news.
Excellent. Thanks, Tam. So we thought it would be helpful to remind ourselves of like the timeline of where we've been coming from with Huntington lowering. So it's almost, again, exactly just over 10 years now since the first people, the super brave participants in these first ever Huntington lowering trials, received the drugs that would reduce the amount of Huntington protein that their bodies were making. And this was at a time where we had no idea how well this approach was going to work. We didn't know how safe it was going to be. It was like being on the dark and scary part of the yellow brick road with flying monkeys around and we're not quite sure what's going on. But along the way, we've been getting, you know, promising signals. And in last year, there was just a slew of so many encouraging and positive, like seemingly positive updates that we were receiving from not just one company, but multiple companies who were all working in this space. It was like the light was starting to shine on the path forward. And then this year, literally great timing on this conference, by the way, because just about a month ago, we received some, you know, news that gave us hope and cautious optimism for this approach and how we can treat Huntington's disease. So it seems like maybe the Emerald City is now within sight. So let's get into that, because I think that's what most people here are very interested to learn about. And, you know, we were kind of as shocked and surprised and, you know, hopeful when we read some of the first news from this uniQure press release, which came out, I think, at the end of September. So I thought that was my phone. I was like, oh my god, I would turn my phone off. But no, we're all cool. So what we think this is like the first domino falling in news that we think is encouraging, not just for uniQure, but for all of these other companies. So let's think about what they actually reported. This data that they're reporting is from 12 people who received the higher dose of this gene therapy called AMT-130, which is a microRNA that gets made in the brain. So it's like the gene therapy, this virus that gets injected by this brain surgery, is now your body has the instructions to make this particular type of drug. And it just means that both levels of the Huntington protein is designed to lower both levels. So your the disease form and your regular healthy form. So for these 12 people, we have data to a 3-year time point right now. And the key thing is that the uniQure is reporting that this trial met its primary endpoint. So this primary endpoint is they report 75% slowing of Huntington's disease symptoms as measured by this thing called the CUHDRS, which is this composite unified Huntington's disease rating scale. And this is lots and lots of different measurements that we make of all different kinds of signs and symptoms of Huntington's disease. So it's like movement symptoms, psychiatric symptoms, but also things that are practical, like how well can you still drive? Are you able to go to the shops by yourself? All of these other things, like how you live your everyday life. And so when they look at that, in their report, they say they see the 75% slowing, which they equate to, and I think Sarah Tabrizi says that this is a bit like symptoms that would normally take a year to happen, or a year progression, will now happen over the course of 4 years, according to this data. The other thing they see is this secondary endpoint. So this is like not the main thing the trial was trying to look at. Sorry, finger pointing these now. But the next thing that they want to look at, total functional capacity, or TFC. This is about the things we were talking about just now, like how are you living your life? Can you have a job? Can you do your own finances? All of this kind of stuff. And these are things that people with Huntington's disease experience get more tricky over time. And so that decline of being able to do those things was also slowed in people who were in this drug. An important thing to remember is that all of this, because when we're doing a brain surgery, it's not fair or ethical, really, to give someone a brain surgery and not deliver the drug. So, this is a lot of, it's a very invasive and difficult procedure, and so they aren't comparing to a placebo, which is the normal way that we would look at clinical trial data. Instead, what they did is they look at all of this data from Enroll HD, which probably many people in this room are participants of, where, this is you go to clinic once a year, and you have all these different measurements and tests done to see how you're doing and how your disease may or may not be progressing. So where they're at right now and what they've told us that they want to do is they are trying to apply for accelerated approval for this drug in early 2026. So you might be like, Rachel, why are you not jumping up and down? And this seems so exciting, we can slow down disease, all of this stuff is happening. And it's, believe you me and Tam are so excited about this, but we're just very cautious about what's happening right now. So only 12 people are in this data set so far, so we've got to be really careful. It could be, maybe these 12 people they just responded really well to the drug, but that's not how it's going to work out when we deliver this to more people. So we just don't know, it's very small. That's why, normally when we get to a big phase 3 trial, if it's an easier thing to deliver, we'd often be giving it to hundreds of people, like hundreds and hundreds of people. So we can see, on average, across all those people, we don't have to worry about too much about someone being a bit of an outlier in one way or the other. So that's why we've got to be really, really cautious in how we look at that. The other thing is that this is only being shared as a press release. So neither Tam or I or any other clinician is able to look at the data yet. So that means it hasn't been through peer review. This is a lot of, trust me, this is what the data says, which, the scientists at Unicure are very good. So I'm sure they've done a good job, but no one else has given it kind of independent oversight yet. So we kind of want to make sure that, that's why we want to open up the data and share it with as many people as possible. That's the normal process. So we can all be like, are we all on the same page? Do we all agree? So we're waiting for that to happen. The other thing is this use of the Enroll HD data. So as the way that we're comparing, is the drug making people better, or what's happening with those folks? And the reason we normally would always use a placebo is because the placebo effect itself is so big. And particularly when you're delivering a brain surgery, this has a huge placebo effect. But we don't have that with this data set. We're comparing to Enroll HD, which is called natural history. So these are people who are not receiving intervention, typically. They're not, in some kind of other arm of the trial. They're just folks who are signed up to this to track how their Huntington's is progressing. The other thing we need to be cautious about is that we want to be very confident that this is working, because this is designed to be a one-time brain surgery, and if that's something that we're going to decide is a good option for people, that's an invasive, difficult procedure which has complications and risks associated with it. We want to be so sure that this is the right thing that we're doing before we tell people that this is the way that we're going to treat Huntington's. So there's a lot of questions that still remain about this benefit that we see. Will it carry on? We now know to 3 years. What about in 5 years? What about in 10 years? And the unfortunate thing is that we just don't have that data yet. And this is just the way that science goes. Science is so annoyingly slow. You can talk to everyone in my group about how impatient I am all the time, that we can't use a time machine to get some of this stuff done quicker. So we'll have to see. The other thing we've got to be careful of is that because we don't have the data, we don't know if maybe they're telling us averages of the data. Maybe one person did crazy good, and everyone else did okay. And these people, we call them super responders. Some people in drug trials, they respond so well. They're all the way out here. So we kind of need to know that, because we need to know on average what's going to happen to everyone when we decide to give them a drug like this. And then the other things we need to flag is obviously how even if all of this looks so great and everything works out and everything is as we hope, how do we roll this out? This is a brain surgery. It's expensive. Gene therapies are typically in the order of maybe 5 million Canadian. It's usually 2 to 4 million US dollars. So per person. So we need to, there's so much work that needs to be done. So okay, now I've just been very, very negative and HDBuzz kill on all of this. So I do apologize. We are very excited, but I think we just have to, as a community, just be aware that this is cautious optimism. Things seem to be heading in a really positive direction, but we also need to hold these companies accountable, and we need to hold, all the researchers and everyone else accountable that we keep things going, we keep the science going, okay? All right, so now we've done that, okay. So there's also some boring admin, I think they're boring admin things that Unicure will be doing, but they are important. So Unicure has been working for a long time. They're committed to working with the FDA to figure out all of these things. Like, how do we've never had a drug before that seems to be slowing down Huntington's disease. So that's so exciting. Now we have all these new problems. How do we decide if the drug is slowing down Huntington's disease enough? What is clinical, meaningful slowing? We're trying to figure out all these new things. It's exciting problems to have. And so they're working with the FDA to try and figure out what all these different things might be. And, they're expecting that they're going to start giving this drug to a lot more people, either in the form of clinical trials, or maybe if it's approved. We don't know yet. But that means they need to scale up their manufacturing. They need to make much more of this so that whatever option happens, they're ready to go. So they're doing all that stuff as well. And they've been working as well. This Enroll HD data set that they're using as their comparison. The FDA is on board with this, and they're kind of defining exactly how they might be doing that kind of nitty-gritty data stuff. So the key things that we're looking at for measurement are this NFL and CUHDRS. So CUHDRS we've already talked about. This is these changes in certain types of signs and symptoms of Huntington's measured in all of these different ways. The other one is NFL. So NFL, or neurofilament light, is a biomarker that changes depending on how healthy your brain is. And what I can tell you is that NFL didn't significantly change in people who received this drug. So it's reported in the press release, but the change is not significant. And this could just be it's just not enough people, maybe it's not enough time. But in some ways, I view this as potentially positive. People didn't get worse. And we as a community have had so many difficult times where things haven't gone as we want. And NFL was a good signal that things were not heading in a good direction. And so this has not got worse. So it's not necessarily positive, but it's not necessarily negative. We'll see how it goes. Right, you've heard enough of me now. I'm going to hand up this time.
All right, so, okay, thanks, Rachel. That was really the thing that we thought that you would want to know the most about. And, we recognize that that news made waves in the community, and it was happy, it was emotional. We both cried, probably a lot of people here cried too. And I just want to acknowledge that that was that was intense. So please, if you want to talk more about it, we're happy to answer questions. And so, okay, so we covered that, but we do want to let you know it's not the only thing going on.
Oh, yeah.
So I'm going to give you some updates from the other companies, just to keep you right up to date. So Roche is running Generation HD2 trial. This is trialing the drug called tominersen. You may recognize that name. It has a longer history. Some of you may remember Generation HD1 trial, which was halted back in 2021 because unfortunately some of the folks who were being treated with tominersen were getting worse. What they found when they went and looked at that data from Generation HD1 was that some people at earlier stage disease might have actually been doing better. And so they had to halt the trial at that time, but they actually made an entire new trial, Generation HD2, in order to formally test whether that group of people who were a little bit earlier on in their progression might benefit from tominersen. And so, so far, it's been safe, and that's good news. The latest news is just a kind of a minor technical detail. It's that the people that so they had two groups. Some people were receiving 60 milligrams of drug, some people were receiving 100 milligrams. And in an independent committee that reviews the data midway, they looked at the data and they said, you know what, if this is gonna show benefit, it'll probably be at the 100 milligram dose. So let's move all of the folks on the 60 milligram to the 100 milligram. So, you know, what does this mean? We want to try and read between the lines. We can't. We don't have the data. We don't know. You know, it doesn't mean that the 60 milligram dose doesn't work. It doesn't mean that the 100 milligram dose does work. It just means that the best chances of finding out if it works is if they use the 100 milligram dose. And so that's a quick update on the tominersen trial. Again, cause for cautious optimism that, you know, Huntington lowering may prove to be an effective approach.
I think this is also exciting, right? Because something, although we don't know if the drug works, we're kind of at the same time point now that we were in generation HB1, where things had to be paused. And we haven't had that. There's no, so this is encouraging that maybe the safety and everything else is better this time, don't you think?
Agreed.
Yeah.
Yes, kind of like no news is good news.
Exactly, yeah.
Okay, so it has been a little bit since we've had an update from WAVE. The last update we received from WAVE was back in the summer of last year. But as Rachel mentioned, kind of like these clues that things might be going well is the, you know, the overall vibe of the update. So what did they show us? They showed that their drug does indeed lower the diseased copy of the protein, but not the healthy copy. And that sounds kind of trivial, like, of course, but it's not trivial. You have to show that your drug is doing what it's meant to do, and a lot of trials actually don't even make it past that point. Yeah. So this was good news. Their drug is doing what it's meant to do to lower just the diseased copy of the Huntington protein. They also shared there may be some clues that, that it might be beneficial in slowing disease, but at that point it's not enough people and it hasn't been long enough to know for sure. But again, it was safe and people certainly weren't getting worse, and so that's good news. And they've said that they plan to go to the next phase, and so we're waiting for news about that. And HDBuzz will certainly keep you posted. The other company that's working on Huntington lowering, this is the company that has preferential lowering of the disease protein. The latest we've heard from Vico is that, again, it does in fact reduce the disease version of the protein. Again, sounds trivial, but it's a milestone. So target engagement, they've done it. It appears to be generally well tolerated. There were a couple of participants who developed a condition called radiculitis. This is a nerve inflammation. They have since recovered, and so Vico has adjusted their dosing, and the trial continues. So as with WAVE, we're just waiting to hear about them going forward with their future trials. Okay, so PTC developed this drug called Votoplam, and they ran it all the way to a phase 2 trial, at which time Novartis is taking over. So, you know, that's also a good sign when a big pharma company is like, I like your drug, and we're like, okay, or we like it too. So in May of this year, we learned that the results of the phase 2 trial for Votoplam, most importantly, it was safe. I did mention, this is one of the pill forms of Huntington lowering. And it does lower both copies. So far, it's been safe. So again, another example of we might be able to safely lower both copies of the Huntington protein. Similar to the WAVE results, some signs that it might be working. But again,
I think I have sufficient caveats in there.
you know, it's like we are very conscious about false hope. We don't want to, get everybody riled up and then have those disappointing results that we've all been through. It hurts. So maybe, might be.
But there's a lot of maybe might be's right now, so
So a larger study is needed to confirm, and that's exactly what we're waiting to hear. So Novartis is taking over. They are planning a phase 3 trial. Phase 3 trials are much bigger. We're going to get the answer, and we have our fingers crossed. So Votoplam, this drug is interesting because it's what's called a splice modulator. It sounds like what's the thing that in Back to the Future where oh come on, somebody knows it. Thank you. Flux capacitor. Okay, Rachel, our next year's capacity Back to the Future.
Okay,
got it.
Have the full uniform 8 to 10 and everything. Great, love
Great, score. Okay, so you gotta stick to
Okay, so on track, yeah.
So Votoplam is a splice modulator, which is just a fancy term for a drug that basically messes with the machinery that produces the Huntington protein from the Huntington gene. And the reason I bring it up is that splice modulators can also be designed against other proteins, not just the Huntington protein. And this ties back to the idea of somatic instability or somatic expansion that we talked about before. Because, in fact, a company called Skyhawk has developed another splice modulator that it targets the Huntington instructions, but it might have the added bonus of lowering one of those proteins that is responsible for somatic expansion, this protein called PMS1. And so we recently learned from Skyhawk that so far their drug, which is called Sky 0515, is safe. And again, it leads to the reduction of the Huntington protein, this trivial thing that matters a lot. And so, so if we have this target against PMS1 as well, that might have a 2-for-1 where we're lowering the toxic Huntington protein, but also slowing that driver of disease somatic expansion. So we will get the rest of the story from their first trial next year. And the next phase is currently underway in Australia and New Zealand.
I would like to apologise to all birders in the audience at this point, as I realise I put a vulture instead of a hawk in the background of the slide. So next time when we have a Falcon HD, I will do better research on the birds that I'm using. So
I didn't notice that.
We're scientists, not ornithologists. So
All right. So the other recent news worth celebrating is that we've learned that the first people were dosed for 2 more Huntington lowering drugs. So we've got a lot of options on the table now. One of these drugs from Alnylam, this is another one of those ones that's administered by spinal tap or lumbar puncture, and another gene therapy. So similar to the Unicure approach, a company called Spark developed this gene therapy, also delivered by brain surgery. It's now being taken over by Roche. And so I hope that we've convinced you by now that there are many, many approaches being taken to treat HD. They're all at different points of the path, but it's moving forward. And so I'll hand it back over to Rachel.
So in addition to everything that we're learning about what's happening right now in the clinic, HD researchers are not satisfied with that. They're always looking to new technologies, new ideas. How can we push the science further all the time? We've got all these great folks doing Huntington lowering, but we're like, okay, but we still need backup plans. We still need other things that we're going to be thinking about to help us get the best drugs possible forward for people. And one of the key things that folks are looking at is this idea of CRISPR. So this is this like clever, like kind of molecular scissors that's able to edit our genetic code. And this is something that only really was first described in 2014. And we've had this explosion in knowledge. It was awarded a Nobel Prize. There's all of this stuff happening. And there are lots and lots of folks working in this space for HD. But the big news is that, in the last year or so, we had our first ever CRISPR drug approved. So this, in the space of 10 years, from initial discovery, we now have a drug approved. Not for Huntington's, but for another type of genetic disorder. And so this drug is called Casgevy. And so this is for sickle cell, and this is now a successful treatment, one-time treatment for people who have sickle cell, which is super, super awesome. The thing is that, not to say that sickle cell is easier, but it is kind of easier to treat because you can take the cells out of someone's bloodstream and edit them and put things back, or you can remove things, put them back. No one is, and this is really important with this kind of therapy where we're editing the DNA letter code. We want to know we did it right and we did it well, and we can have all these checks and balances. The problem with Huntington's is that we don't really have a safe way yet to know for sure that we can actually edit in someone's brain, which is where we want this kind of stuff to go. And that we've done it right. So we have like a lot of extra barriers in Huntington's disease to try and get this kind of therapy working. But that doesn't mean that people aren't trying, and there are super clever scientists all over the world who are really, really working on this. Oh yeah, that was a great animation.
Rachel, wasn't it supposed to scroll?
It was definitely meant to scroll up. Basically my PowerPoint skills
There's
are
lots
really
more
lacking.
down below. It's okay.
Yeah, so if you can see the full slide, there's about 20 studies that you can't read on this slide apparently. But there we are, never mind. And so people are designing all these different ways. How can we deliver this kind of drug into the brain? How can we get it to spread through the brain? How can we be sure that we can edit the Huntington's disease mutation in a way that's safe, it doesn't cause off-target effects, and it gives us the result that we want? And at what point would we do that? And there are people all over the world. People are really interested in this. And the reason that people who work in this technology are so interested in Huntington's disease is because it's a clearly defined disease. Everyone who has Huntington's disease has the same kind of mutation. But also, it's because of people like you, your organization, your advocacy, like your commitment to natural history studies and so forth. Being an organized community is so attractive for people who are looking to apply their technology for this kind of thing. And so that is why these companies are really interested in Huntington's. And there's two that are kind of leading the way in the Huntington space. One is Incisive Genetics, the other one is Life Edit. And they are attending lots of the scientific meetings and clinical meetings, and so we're really hopeful that sometime in the future we might learn some more about how their work is going.
Oh, so we thought it might be worth just briefly going over advances in biomarkers, because biomarkers are important for all of these clinical trials to be able to move forward and be able to know whether they're being successful or not. So what's a biomarker? A biomarker is any measurable clue that tells us what's happening inside the body. And so it helps us, predict, track, and respond to the disease before later outward symptom onset. And so these tools are super important, and we've had advancements on this front as well. So the first one, it's been mentioned because this is the thing that companies are using to measure whether their drug is working. And so it's called neurofilament light, or NFL. And when neurons are stressed or injured, they basically shed bits of themselves, and NFL is one such piece, and it floats through the spinal fluid and even into the bloodstream. And so this is a measurable thing. In HD, the NFL levels go up gradually, but they start to rise years before outward symptoms show. And so this is a very good measuring stick to see whether disease progression is being slowed by a drug. Another important measure is the disease protein itself, expanded Huntington. And so advances have come along so that scientists can measure how much of this disease protein is in our spinal fluid. And you've heard now about several clinical trials that are trying to reduce Huntington protein, and so it, again, seems trivial. We need to be able to measure if it's going down, and thankfully there have been advances on that front, and so we can actually measure whether the drug is doing what it's meant to do. Also coming down the line is machine learning and digital tools. These things are amazing. They can spot patterns in speech, movement, and even like phone usage. And so this is way easier than getting a spinal tap. And so this will definitely make clinical trials easier, smaller, and smarter.
So we want to end by thinking about, and reminding everyone in this room that, we've got these clever scientists and clinicians, but the heart of HD research is you. Like, it's everyone in this room, it's everyone who's ever read a paper, who's signed up for a trial, who's ever donated blood or spinal fluid. None of the work we've talked about today would have been possible without participation from the community.
Not to mention the folks that are driving their loved ones to
one of the questions we get asked at HDBuzz all the time is like, well, what can I do now? Like, what kind of actions can I take? Or, maybe pointing people to different resources. So we wanted to kind of end by showing you kind of cool things that folks can get involved in. So we talked about, there are obviously the clinical trials that we've mentioned, but there are all these other kind of, we call them non-interventional studies or natural history studies, which are looking at people and how they progress through disease. And this sort of stuff is so important. So if we don't understand exactly how disease is normally progressing, how will we know if the drug is going to be changing that? And one, so we have Join HD. This is something that's really important for juvenile Huntington's disease, which is, even rarer than adult onset HD. And so just being able to know how many people out there have juvenile Huntington's disease is a really important thing to be able to know and understand. It gives us the right to advocate for more money, resources, clinicians, and other bits and pieces. So this sort of stuff is really important. The other study I want to talk about is HDYAS. So this is the HD Young Adult Study being led by Sarah Tabrizi in London. And this is such a cool study because what she's doing is she's looking at folks who are gene positive, but often decades before they're predicted to start having outward symptoms. And what she's trying to look for is trying to understand, are there any small signs in these biomarkers, like what Tam was telling us about, that can kind of give us an indication of that disease is happening, or that things, what path is this person on? Can we predict all this stuff? And this really matters because our ultimate aim is to treat people in the long term. We want drugs which are going to slow, halt, or reverse disease, but we want to give them to people before they've even had symptoms. So if we can even prevent the symptoms happening, that would be great. But we can't treat people who are really, we can't run the clinical trials in people who have no symptoms and no signs of Huntington's disease for us to measure. So we have to find those signs. We need to know that those signs aren't changing or how they might be modulated by the drug that we're looking at. So this is like a super important thing that's going on. Similarly, there are all kinds of different surveys. I highlight the ones here from HDO, but most Huntington's disease organizations around the world have surveys where they're collecting all kinds of different data about, some of it is about how these organizations can better serve you. Some of these are scientific surveys, all sorts of bits and pieces. And then the one we want to talk about really is Enroll. So I'm going to hand this over to Tam.
Thank you. Some of you probably have already participated in Enroll. If you don't know what Enroll HD is, it's an observational study. So basically, you go to the site once a year. They do some tests. I've been going since the outset. I think this might be my 12th year next year, and I still can't do those cognitive tests. They ask you things like, say as many words as you can think of that start with the letter F. I can't get past the first one. No, but basically they do these tests. They do like a neurological test. They collect information, and all that information goes once a year into this huge bank of information. So valuable. Because of the participation of the HD community, there are over 20,000 people that have been going all of this time. This information is being used for things like the Unicure trial. So if you've participated in Enroll HD, you've contributed to the Unicure trial. And so this is when Rachel was saying the HD is an attractive disease for companies to look at. This is one of those organized community things that she's talking about. So yeah, very important in clinical trial design. The data can be used in so many ways. It can also be used to see, are people taking one kind of drug? Do they, is that associated with slower progression? Or even finding new targets based on the DNA from all of these samples. So, so Enroll HD. Yeah, if you go to the website, if you're not already enrolled, if you go to the website, it's a very straightforward process, and I highly recommend it. Is this me too? Right. So, Enroll might be the biggest study, but it's not the only observational study. There are many options. Lots of these things are on your local organization website. In North America, HD Trial Finder is a fantastic resource for just finding out what's out there. Not just the trials that are testing drugs, but also the observational trials, the surveys, everything. So you basically just put your information in there and say, I want to search within a 100-kilometer radius, and it'll tell you what's available in your area.
Yeah. So if Ed Wild was here, he would tell you one of the best things you can do for yourself if you're living with Huntington's disease is all the really boring things that even regular people and everyone else, we know really what to do. And actually, me and Tam did a very bad job of two of these things last night. Had probably a little bit too much wine. We haven't seen each other for a little while, and didn't get to bed as early as we were eventually. And so some of the key things we could do, we're learning more and more from all kinds of studies that actually just leading an active lifestyle, doing exercise, all those things that we're eating well, not drinking too much, not taking too many drugs, or any drugs. And just getting really good sleep. All of this stuff is really important and actually can make a really big impact in how people experience symptoms, how symptoms might progress, and all sorts of other bits and pieces. So one of the other ways that So one of the other things that some people might be interested in terms of how they can contribute to research is to think about, when you in the future, you might want to consider brain donation. And so this is something that the Huntington Society of Canada is able to facilitate. There's actually folks, the folks from UBC are here for the HD Biobank here today at this meeting. And this is one of the most selfless and amazing things you can do for HD research if this is something that is for you. It's not for everyone. And Tam, this is something that you've been thinking about doing, right?
Yeah, I just wanted to mention because it's not something that once you decide, which is a very personal decision, it's not something that takes a lot of work like in the moment right now. But it has to be organized if it's going to be carried out properly. And so I highly encourage you to talk to the UBC folks at the table. Basically, there's a form, you fill it out, it's a consent form, you read it, and if you consent, you sign it, and you tell your loved ones, if something happens to me, this is in this filing cabinet, make sure you get it, because it's basically like a 12 hours, they have to get that brain out. So, yeah.
Yeah, and so this is something you've done.
Yes, so I put we're a little bit morbid sense of humor in our house, so I just put a sticky note on it that says, if I die, call this number.
There we go.
And told everyone where it was.
So the reason we want to highlight this is in the last year or so, we've had what are considered some of the biggest landmark studies in terms of our understanding of Huntington's disease, exactly what's going on in people's brains and how things might be changing over time because of incredibly selfless people who have donated their brains to research. And they're just highlighting some of these here. This is where we've really begun to understand exactly what might be happening with semantic expansion, all these kind of other drivers that we think could be really good ways to develop new drugs for Huntington's disease, all enabled by these brain donations. And it's just really spectacular. All right. So finally, we just want to flag some of the other resources. Obviously, we're at the Huntington Society of Canada meeting, and these folks have so many resources for you. Like, their website is full of all kinds of information. Tam already mentioned HD Trial Finder and the Huntington's Disease Foundation as well. These are all folks who are putting on research webinars and all sorts of Q&As with different companies, and it's really, there's so much resources out there, and we encourage you to find those. Because keeping yourself informed, whether through this or through HDBuzz, this is advocacy. Like, if you are empowered by knowledge about what's happening in this community, you are pushing the needle. So this is something we just want to say. You're already doing it just by being in this room, which is super awesome. And this is something that, this is a community effort. We, as researchers, as clinicians, as whoever else, we have to work together to push things forward to get us to the Emerald City with our disease-modifying therapies. And we're so happy to be here because for Tam and I, this community, this is our home, this is where we feel so happy and comfortable, and I'm so happy that we've got to be able to talk with you today. So we'll end things there, and we're happy to take any questions.